have been working on bird flu, H5N1. They've been working with ferrets, which are the closest thing to an animal analog or the way it behaves in humans. And they have developed what looks like a version of this particular bird flu, which may transmit more easily between humans. And the American Federal Biosafety Board has immediately brought in held hearings to decide how much of the details should be suppressed. The thing that amused me was that the very initial reports presented the work on the ferrets as essentially serial passage. You pass the flu from ferret to ferret to ferret until the ferret in the next page over gets sick. This is not rocket science. That cat is out of the bag. Serial passage is also used to weaken viruses, as in the live polio vaccine. And my thought is if a cat of serial passage gets out of the bag and somebody remembers polio vaccines, and somebody thinks of Ebola, which is not a problem because it kills people too fast, a little bit of serial passage work might weaken it just enough to survive an airline trip to an airport. And then something that really happens. And shall our bacterial experts comment on this particular dreadful scenario?
Then you have the last slide of Dr. Ain, last slide of Dr. Ain, raise your hand if you have any idea what we're talking about. The old story by James Tiptree Jr. from about 40 years ago, where a guy goes around airport to airport to airport, and at that time you could actually go outside. So when he's in the lounge, the story goes outside, he feeds viruses to birds all around the world, with the idea that the birds will then spread the virus to humans and then remove the cancer of human beings from us.
Well, if you believe overpopulation is an issue, okay. There have also been stories written where somebody goes to the airport and there or something.
Very
Yeah.
Well, those stories are very dramatic. At the same time, from my perspective, what happens in real virology is even more dramatic than that, because you have to imagine all the enormous reassortment of these flu chromosomes that's going on all over the world, particularly in the swine pens and in the flocks of birds. And the flu virus is unusual in that its gene has 8 separate genes on 8 separate chromosome segments, and they're just floating in a bag that makes the virion, and it just packages them at random. So that's why you can have a cell co-infected by a human virus and a bird virus, and they just get packaged in the same little bag with a virion, and all of a sudden you have a new virus that's more virulent to humans and birds.
Also, why we have a new vaccine every year.
Yeah, multiple new vaccines. So in fact, for often the annual flu vaccines don't even work because you have to guess at what the new strain is going to look like. So my feeling about the ferret research is I wasn't sure why it was such a big deal, because whatever's going on in your lab with ferrets, there are these humongous swine farms in Hong Kong, in central swine markets, where they now test looking for new flu variants, figuring that's where they're going to appear, because that's where all this natural mixing occurs. So I think that if the virulent avian flu is going to happen, it's going to happen on its own, regardless of what people are doing in the laboratory.
So in other words, what you do about lemons?
Well, no, I'm not a virologist per se, and I know a lot of virologists are worried about this, but as a microbiologist, I'm kind of wondering why they're so worried about it. What gets lost is why they're doing the experiment. I mean, does anybody wonder why they're doing this experiment, right? Okay, well, and this is always parodied as, because I can. And so, what the real reason why you do this is to help the people doing the sampling in the Hong Kong market and elsewhere, to help them know what to look for. What are the specific genetic changes that lead to increased virulence, So the idea is that if we know what those changes are, then we can be on the lookout for those and maybe find the genetic type before it gets out into the whole population and be ready for it. So I think that research is valuable to do, and it's not easy research to do. I mean, growing and culturing flu virus is pretty sophisticated stuff. It's also not a good terror agent because you don't go to paradise by dying of flu. So I really don't think I don't understand why it was necessary to censor that. I can see controlling it and doing it in biohazard facility and all that for sure, but to censor the knowledge, that really seems questionable to me.
The Reuters report that came out yesterday announcing the final agreement that the papers will be published with the details suppressed. And this is current events.
Except for 1,000 people out there who need to know.
Right.
1,000 best friends, I mean, that's kind of silly.
And as I was saying, the early press releases basically let the essentials out. But according to the Reuters report, and some of the researchers, such as Bob Cushier, the Dutch researcher, are saying, as Joan is saying, that the problem here is nowhere near what people are worried about, simply because you do get this natural decontamination.
Yeah, I mean, it seems that the politicians are often a couple of steps behind the scientists and worried about stuff that doesn't really matter. When in the early 70s, when we were first starting to do genetic engineering and moving genes from humans to E. coli, like insulin, things like that, there was a lot of worry about these bugs, bacteria, getting out of the lab, and ultimately the decision was to use certain strains of bacteria that theoretically could only survive in the lab. So for instance, they were not able to make a certain nutrient, or they required a particular amino acid like lysine added to the media in order to grow. Michael Crichton actually picked up on this idea in Jurassic Park. The dinosaurs, the genetically engineered dinosaurs there cannot make lysine. And so lysine is an essential amino acid, so theoretically they shouldn't be able to escape from the island. But then,
Yeah, that makes sense.
so, and then his idea is that the dinosaurs learned that they can eat bananas, which turn out to have a lot of lysine. I didn't know that, but
In terms of taking that a step further, for a science fiction piece, I guess one reason I find it hard to take this seriously is that I'm already looking to the next stage of things to worry about. So in my book, The Highest Frontier, the problem with flu is that we now have printers that can print out stuff. And you've all heard of 3D printers that can print out constructed stuff. So we now have printers that print out stuff at a molecular level. And so the computer viruses of the future are real human viruses that print out. So you can literally print out flu virus particles out of printers. And so the student dorms get all these flu viruses printing out of their printers, and that's how they get sick. And I hope somebody's thinking about that, because that is likely to be happening eventually.
Well, you introduce yourself saying that among your research in the lab or directed evolution, you are aware of the work that was done in the last few months, or published in the last few months, on making yeast multicellular.
Well, making yeast multicellular, actually, that's not, I hate to,
What I wanted to ask next is, where do you think that might go?
Yeah, well, first of all, the trouble with being a microbiologist is that things that happen in real life microbiology are more amazing than anything you do in the lab or even in science fiction. So yeast ancestrally were filamentous fungi, but they evolved to be unicellular. So they still have the pathways in there. I didn't think it was actually that significant, because in natural world, single-celled yeasts all the time evolve to be multicellular. Multicellular evolves to be single-celled. It just depends on where they're growing many generations. So yes, well, directed evolution, this is done all the time now. I'm sure that in Novartis, you're doing this already to to fine-tune your strains. So this was what my talk at 11 was about, was directed evolution. So you can do directed evolution as a lab science. Richard Lenski was the first to do it, and he got slammed by all the anti-evolution people because evolution is in the lab now. Hey, we can do it. We can predict how long, how many generations it takes a tree to evolve, and and it's being used in industry as a way of making better products. So now in terms of evolving multicellularity, that it turns out that multicellularity and unicellularity are not as far apart as people thought. You mentioned yeast evolving to be multicellular. Well, yeast have genes in them that are homologs of breast cancer genes in the human body. And when you take those genes, you can take people that have a breast cancer homolog gene that's cancerous and put it in the yeast cell, and the yeast cell proliferates. That's a test for a certain allele of breast cancer. So actually, that's something that, that's coming up.
Well,
For those of you,
experiment. What was really interesting to me about this experiment, they got these, they're trying to figure out how did single-cell life become multicellular organisms. And so they got these yeast, single-cell organisms, to clump together and to begin to change their behavior so that the yeast cells in the inner part of the clump would do different things than the yeast cells in the outer part. The outer ones had to interact with the outside environment, and the ones inside had to do things to transfer, nutrients and other things to other cells. So they began to take on specialized jobs within the cluster. And I want to just read you a quote from the article. It's really interesting. When the clusters reached a critical size, some cells essentially committed suicide to allow offspring to separate. They make the cells in the cluster make sacrifices for the common good and adapt to change. And that's an evolutionary transition to multicellularity. So I was thinking about, so they say, in the human body, all of your cells are cooperating in order for a sperm or an egg to get out there and to reproduce. They're not going to be reproducing themselves. They're stuck in your body, but they're going to perform their roles so that you can, as a multicellular organism, reproduce. And I was thinking about our planet as a bunch of multicellular organisms that are perhaps clumping together into one gigantical cellular organism, and that some of us have given up the right to reproduce so that others may, so that we don't overrun the planet. I mean, in a way, you can think of our behavior as changing in this global community into being a multicellular organism, taking on specialized duties and so forth.
Super organized.
I thought that was interesting.
Is there that much difference between what they're doing with yeast and the biofilms and bacteria? So for example, like when you have a sheet of bacteria, not as an individual itself, but as a sheet lining, say, the inside of a water pipe, where the ones on the outside kind of act as a physical barrier, whereas the ones that are buried and not exposed to the toxins and stuff.
That's exactly right.
Multi-species combination.
That's multi-species, that's right. In bacterial biofilms, in effect, those, you can think of them as either a community of organisms or as a multicellular entity, even though they're multiple species, because there'll be the ones that are underneath, like the stuff on your teeth, okay? Whenever you get the white stuff that gets picked off your teeth by the dentist, that's biofilm. That is a multicellular bacterial entity, and it has channels through it for nutrients. It has ones that use anaerobic respiration or aerobic respiration, things that bind together so that it puts out these long stringy chemicals that hold it all together. And biofilms are a huge deal. And what do we call this? Do we call this multicellular or unicellular? It's a matter of definition. It's a very exciting industrial field because some industrial energy processes work best through biofilms. At the same time, it's a huge environmental headache, and so cleaning up biofilms, one biofilm can want to clean it out. It's a huge, interesting area.
These viruses, phages.
Biofilms tend to be phage resistant. They're also going to be antibiotic resistant.
Okay.
So that's the one of the reasons that we worry about, like on catheters.
I do have one.
They can't percolate through all the layers of the cells on the outside, so the ones
I do have one plastic tooth, which every so often we biofilm and pump up with it. I think Clorox is nice. But you have to get things with pretty potent combinations of reagents. Next question. The field that I wanted to bring in for our discussion here is synthetic biology. This actually has some history to it, but it hit the news in a big way last spring when research crew says, okay, we have assembled the chromosome of a bacterium from veg chemicals. We have built the DNA from scratch. We didn't make up a recipe, we just copied the recipe, but we built the DNA. Then we took a cell from which we removed the DNA and put the synthetic version in, and lo and behold, the cells came to life, multiplied, everything's working. Which was a neat trick. Some people like it as saying you keep the Coke can and you make up some Coke and you pour it in and say, see, I made Coke. Okay. But some of the discussions say, look, if we can do this way, the next step is going to be not just to copy an existing chromosome, but to do some editing in the process, or do some synthesizing in the process. And you can build a bacterial cell, a yeast cell, maybe even something more elaborate with some real interesting tailor-made qualities. And I don't know, somebody who works with patents, that should be real interesting.
Yeah, well, one of the things that we worry about a lot, because I was thinking about this after you brought it up, and I was thinking about, like, why would you want to do this besides just, like, because you can, because it's cool, and sometimes you can just because it's kind of neat. But oftentimes when you have a cure or a drug that you want to administer to people, you have to make lots of it. So the question is, how do you grow it in bacteria? Do you grow it in animal cells? And there's a big difference, because the bacteria cells will always get a very, very small amount from the cell that you grow it. You just can't get rid of it. Like, maybe it's 1 out of every 10 billion particles. Still, if that's a toxin from a bacteria, and there's a tiny amount of that in your drug, it's still bad. I mean, the FDA is still approved, and
So you're saying you'll synthesize something that doesn't move that?
yeah, exactly. I mean, there are 4,000 or so genes in the drug, right? And what is the number that you can chop it down to? How few genes can you get and still have the cell do everything?
The same people that have been working on synthetic biology have another project that they've been working on trying to establish what the minimal cell is.
Yeah, exactly.
And,
And so again, you have to close the loop. Like if you have or engineer the synthetic cell so it's not producing these toxins, then that would make it a better thing.
it doesn't interfere with their
Okay.
area. Joan?
Yeah, so I think that from a technical standpoint, that was more of a technical tour de force that, as you said, has real making a cell from the basic parts. As I see it, it just adds yet another technical thing to the toolkit for synthetic biology. And what I'll be interested to see is what it actually gets applied for. So you can make genes from the test tube now. But what kind of genes are you going to make? And what is really interesting is to see where genes come from and then what you make out of them. For instance, there was a company, Genzyme, where they took this thing called hyaluronic acid that was made from rooster cones and so on. It's very hard to find, but it has really good uses in the body for surgery and so on. And so they found a bacteria that makes it as the bacterial capsule. It would grow bacteria to make tons of this simple molecule. So the question is, will that become easier and faster to do now that you can snip genes? And I guess Frank would know about this, but my guess is that it's like all of the above, that in a real industrial laboratory, you're going to do the high-tech stuff with the gene, synthesizing the genome, but you're also going to try just growing these bugs and see what they can do. I mean, we still send people out to the rainforest to try to come up with unusual bugs, right?
Yeah, I mean, there are literally millions of bacterial species out there that we just don't know about because we can't grow them in the lab, or maybe they reproduce really slowly. And the doubling time of your organism, the lifespan of your organism, is directly proportional to the time that it takes you to get a PhD. As a bacteriologist, I could think of an experiment on the way into the lab, start my cultures growing, and if I was there for 10 hours, the cultures would be dense enough by the end of the day that I could do the experiment and have my answer.
He always does.
Yeah, it doubles every 20 minutes or so, depending on if you get to work nutrients. Oh, but the other thing that I forgot to mention in terms of whether you're going to make your drugs and protein in a E. coli cell versus a mammal cell, like, there's a lot of what's called post-translational processing. So, for example, a protein may need to be glycosylated, meaning that particular sugars are stuck onto it in particular locations in order for it to be active as a human being. And the E. coli may not be able to do that, but the mammalian cell might be able to. So, and also the other thing with E. coli is you can possibly get sepsis, which is your immune system going crazy because there are E. coli cell wall fragments floating around in the blood, and your immune system just goes insane and kills them.
Well, I guess I was wondering if there's another topic that people are interested in. Where are we going with using the microbes and the synthetic biology as models for nanorobots that are going to go into people's blood system? And isn't there some work now where we're combining the bacterium with robotic parts, and then we can get things that can actually go within the patient? So we're not just making a gene product now, we're actually making entities that go into the body and move microbes.
We're getting into very interesting things like that. In fact, just before I was in Slonczewski, talking about a DNA bot that can activate cancer cells. This is kind of interesting. But to go back to the synthetic biology, before they ever did this kind of bacteria itself, there had been some work done in putting viruses together from off-the-shelf chemicals. And they worked on polio virus, and I think they made smallpox virus. And that should scare people, because hardly anybody these days is interested. There's not enough vaccine for any severe proportion of the population.
Well, actually, where smallpox is going to come from, maybe this is slightly off topic, but where smallpox is really going to come from is natural evolution from monkeypox in Africa. Because smallpox vaccination conferred partial immunity to monkeypox, and now that nobody gets vaccinated anymore, people get monkeypox, and meanwhile, exotic pets bring monkeypox to the United States.
Now the hand is up again.
We were talking briefly about combining robot parts with bacteria or something. How exactly would that work? Maybe you will elaborate more on that idea.
And this involves a form of synthetic biology that's more interesting than just making a gene. Okay, so can you make bacteria that produce? First of all, bacteria actually are nanobots already, because bacteria have rotary propellers with them, and they have rotary motors. Their ATPase motors are rotating. So they have these mechanical things on their surface already. Now, supposing you genetically engineer them so you make a synthetic, if you will, bacterium that has these rotary parts that can now attach to robotic parts, okay, then nanoscopic parts. Well, now the bacterium can essentially power a nanorobot, okay, antibody-antigen interaction. Self-assembly, exactly.
Big magic word.
That's the great thing about bacteria is they are self-assembling.
So when I started my PhD, I was working with a guy named Professor Pate, who's since passed away. But he was working with living bacteria, which, so the deal is that in the cell wall, there's a protein mechanism that spins, and then you have the gel that sticks out, and that acts as the propeller. But in the living bacteria, you only have the protein part, and you've lost your flagellum. But it causes the cell wall in a certain area to spin around.
More like a conveyor belt.
Yeah, kind of like a conveyor belt. And so part of my experiments in my first lab rotation was mixing beads that were big enough to see in the microscope with the cells. And you can see the cell here with the beads spinning around are like rotating around the cell.
Here's the cell. And here's the bead. And they move. I don't know how well anybody can see it. I mean, a little piece bigger. But it's something better than that.
So yeah, the cell will be like this. The bead are a couple of beads, like 5 or 6 beads in the clump. They would just rotate it around or like stick on the end and spin around. So you couldn't see the flagellar motor itself because it's too small, but you can see the beads spinning.
We forgot to mention that monkeypox had already produced an infection of American prairie dogs that in turn bit people who got monkeypox. So we're very, very much at risk. The Department of Agriculture was panicked in that epidemic program.
Here's a theoretical question. So there's a reserve of smallpox in the United States.
Yes.
Atlanta, as you guys all know, and another one in Moscow. And there have been calls for them to be destroyed. But other people say we should keep them in case we need to make a crap load of vaccine. And other people say, oh, yeah, it could get out, and that'd be really bad. So what do you say, keep or destroy?
Well, one of the arguments for destroying says that all we really need is the string of bases that would allow us to recreate the DNA at will. And nobody has actually tried that yet. Close to it. They've done it before. Okay? Certainly, if all you have is the DNA, you can probably make sufficient bits of protein to get a vaccine going. The thing that worries me is that there's an awful lot of ancient smallpox victims that have been put on ice effectively, buried in permafrost grounds. Permafrost is melting.
Yeah, I think to me, I'm always wary of arguments to destroy. I don't know how you feel about this, but destroying something that exists. I always remember Mr. Spock's comment about the port zone. To destroy a species is a crime against science. And I think there's a profound truth in that. I think we should be very wary about destroying something that we have. Of course, things go extinct in nature all the time. They don't need we're destroying things without realizing it. But where we have something, I worry about that. I think we're reaching the point where you're correct that just having a sequence where information is reality, reaching the singularity point where it's really a moot point because, it's like, exactly, in the highest frontier, this would be a moot point. Who cares if, you can always print it out. We're not quite there yet, so I'd say maybe we're not quite ready to destroy it. The other thing, though, I'm wondering is why do we need the whole virus to make the vaccine? It seems to me if your vaccine is the issue, we make now third-generation vaccines that have never seen a virus. Like, for instance, people don't realize the Gardasil vaccine for papilloma, for HPV, that's never seen a virus. It's recombinant proteins that make it out of. So it seems to me we should have a third generation vaccine for things like smallpox, so you don't have to make, but of course, then you need a whole program to make that kind of vaccine and who would test it. There are all problems for that. So I think it's a tough issue, but I'm hoping that we'll reach the point where we don't need to keep the stocks. I don't think we're there yet. I don't know, could Jeanne weigh in?
I would much rather have the virus in a vault somewhere than have the information about the virus on a computer somewhere.
It's a lip box to reason.
Well, Jeanne, can you say more? What do you think about some of these other questions of, what if it does get out there because people aren't getting vaccinated? Or should we all continue to get vaccinated against smallpox?
I'm all for continuing to get vaccinated for everything.
Well, but what if there's a 1 in a million, what if 1 in a million dies now from the complications and nobody gets the illness? Then if hundreds of millions of people get vaccinated, there's like hundreds dying, right?
Well, I would like to see the vaccine get to the point where it doesn't kill anybody.
Well,
To me, the dangers are so extreme of what we're talking about. You know, somebody creating a virus from scratch, or altering a virus so that it's more easily transmissible, or so that it's more dangerous to us, so that it's harder to kill, or whatever. All of these things are becoming so much easier, and the number of people who can do them are becoming so much greater that it seems nearly impossible that something like this that's very bad will not happen. And before people flew airplanes into towers, everyone thought nobody would do that except for Tom Clancy. And before people started sending anthrax around in envelopes, everyone thought nobody would do that. And so this is clearly going to happen. It's just a question of how and when. And so being as well prepared as we can with our very inefficient system and government is really important, I think. And so I would rather be vaccinated for everything under the sun, even if I suffer for that, and have a reduced risk of things.
The business about vaccines killing some people that receive them is why today many people refuse to let their kids be vaccinated because they think vaccines cause harm. Things like whooping cough. And what do we get today? Maybe 20 people around the country might die from the vaccine, and people have forgotten because so long since whooping cough was epidemic, but the death toll from the disease was up in the 20,000 per year range.
Actually, in that case, I think it's not quite 20, it's a lot less than 20. But you're right, that is the dilemma of vaccines. And if you think of older ones, it's worse with polio. Like, the Rotary Club polio vaccine that's sent around the world to third world countries, that does kill some people. That is a very imperfect vaccine. Whereas the one that we take here is more perfect, but less effective than the one that's wiping out polio. There's a real dilemma. But there are also false dilemmas, like the papilloma vaccine. That is the Gardasil. That's a false dilemma. It's a completely manufactured, political, emotional false dilemma. That vaccine is not killing people, not making them retarded and all that. And so you have it's a real issue because there is a real ethical dilemma with vaccines, but there is also a manufactured dilemma there, and there have actually been, the autism paper was retracted. So it's very hard for public health workers to sort out the real dilemma from the manufactured dilemmas. It's a sad thing.
Most people don't understand statistics.
But the other thing, when you raise the smallpox, I do have to get back to the monkeypox thing because, so often what we worry about is not the most likely source of where things are really going to happen. That's a common, I always have that thought in my book that everybody's worried about X, and instead it's Y that comes up and bites you. So, for example, the plague, plague stocks. There was a case where someone was working with a stock of plague bacteria that was considered safe, but he got sick and died of it. And you're wondering, well, how could you get sick and die of this altered, supposedly safe strain? Well, it turned out he had a genetic defect that had higher levels of iron in his blood, and so the disabled strain could grow now because it didn't need to acquire the iron. So this, with smallpox, you know, we think monkeypox is just an irritant. But you know, where do you think HIV virus came from? It was something that caused mild problems for apes, and then it's turned out to be a big headache for us. So I think that we need to. The take home is we need to worry. Not, you know, at least the smallpox stocks. We know that's something to be concerned about, but we don't think about viral diversity and the diversity of viruses. We need people monitoring those things because that's where the next plague will probably come from.
Actually, one of the things that some people feel that you're talking about is that you can expect more things like Ebola, AIDS, HIV, because these things are coming out of reservoirs in areas where people have not lived, and people are moving into these areas and getting exposed. And some of the stuff they get exposed to has a really nasty bite to it, and there's going to be more of it.
It looks like they're coming up with some kind of possible treatment for Ebola with a small interfering RNA.
Yeah, that was very interesting. That's a really space age treatment where you have RNA molecules that interfere with the Ebola virus. The places where we still see the worst emerging viruses are places where there are a lot of people in proximity with a lot of closely related animals. Okay, so Africa is the most obvious because the chimps and the other primates are most closely related to us. So when it comes to viruses, that's where you find them coming. But you know, it turns out that with respect to influenza and SARS and some of these other respiratory things, I guess pigs are related enough and birds are related enough. So now it used to be the flu that was called the Spanish flu. It actually should have been called the Kansas flu because it probably came from the best evidence suggests that it was poultry farms in Kansas that came from. So yeah, right. So we have to be careful when we say where these things are coming from. But humans together with animals in large numbers, that's where you find the most emerging pathogens. Yes.
Can you talk a little bit about what do-it-yourself biology might lead to, and maybe not in the near future but in the far future, how biology might become a little bit more accessible and how that might change the way we think about biology?
Well, I wish it would change it more. I wish there were more do-it-yourself biology. You know, as a high school student, you can pair up with a lab and do amazing things now. Increasingly, one of the exciting trends is that in government research funding, research grants now all have a component where there's required to be outreach. So the researchers are supposed to be spending some of their grant funds working with kids.
Well, I know one researcher grant proposal would not get funded. As a byproduct of my research, I will invent a little giant home genetic engineering kit.
Well, those exist already. Sorry to disillusion you, but you can clone things already as far as
That's what the African violets are.
But and online, if you want to do a science fair, like what used to be called the Westinghouse Talent Search, but anybody can fill out the forms online and give you the thing to do it, and then you can find your local researcher to help you out,
You
figure
can
out a
find the
project.
equivalent on eBay if you have money.
I'm sorry, you had.
Sorry, what are your thoughts on personal having your sequence by centralizing slide?
We're down to the $1,000 deal.
That's actually the next thing I was going to talk about because I think one of the next major effects is going to be people having specific sequences that are important. So, because so many of the disease treatments that we have only work if you have the right genetics. So for example, Herceptin, which is a huge breast cancer drug, it's an antibody and it binds to the HER2 gene, which to the HER2 protein, which is a growth receptor. And so if you have a lot of these growth receptors on your cells, then your cells are growing a lot. And if you don't have that, you have a different kind of breast cancer, and the Herceptin, which attacks that receptor, won't work for you. So you have to get tested for that. Also, you mentioned siRNAs before, which is actually what I call the second year of my day job, is siRNAs. And what you have is you have a double-stranded RNA, a short interfering RNA, siRNA, which is delivered, which is the problem. Delivered to a cell, and the thing is, human cells don't normally have double-stranded RNA. Exactly. They react against them because sometimes double-stranded RNA is viral, and you have proteins that chop these things up into little pieces, about 21 to 23 base pairs. And then there's another protein complex called the RISC complex, which binds these, and then it finds the corresponding messenger RNA, which is probably the virus, and then chops that up so that you can't make more virus. So if you stick in, so for example, if you have a kind of cancer that overexpresses a particular protein, you can take an siRNA that targets the messenger RNA that corresponds to that protein, and then it will chop up the messenger RNA. But the thing is that it's very sequence specific, and here's where we get into personal genomics and having your own sequence. You need to know your sequence matches 21 to 23 nucleotides or base pair long siRNA to get maximum effectiveness.
I would say the biggest problem in general with getting your sequence is just that you're going to get a lot of data and they're not going to tell you much of what it means. Only a little guidance about what it means. So you're going to get all of this data and you're not going to know anything basically. And what they can tell you is not very much at this point. I mean, scientists know more than what you would get by paying your $1,000 and getting your sequence. Let me just mention one other thing before I go. There's also the issue of, do you find out things that are going to change your behavior in a negative way? Okay, say you find out something that's going to change your behavior in a positive way, like you have a predisposition to breast cancer, so now you can treat it, you can do something about it, that's great. Say you find out something that's going to change your behavior in a negative way, say you find out you have genes that predispose you to alcoholism. So you're just going to give up and become an alcoholic and say, well, I can't help it, I've got the genes. Or you find out your genes are for somebody who's not super intelligent, even though you're in grad school at MIT, and so you say, well, I'm never going to be as smart as these other people, so I might as well quit. I have a friend who I grew up with, my age, who has Huntington's disease, and we kept urging her as we were growing up. Her father had the disease, it's inherited, it's one gene. Kept urging her to have the test to find out if she had the gene or not. And she didn't want to have the test because she didn't want to know. And the doctors sided with her and said, look, lots of people who have this test despair. Many of them commit suicide. Many of them give up on their lives. It's better to just find out when it expresses itself. And she's down in her nursing home and crippled and can't talk. So now we know. But she had some good years of life there without it. So there's a lot of issues.
One more study to mention before we let people tell us that we're running out of time. There was a little while ago a study that was done with mice, and I mention it when I'm teaching because we're talking about the blue. The one question is, what on earth would be the selective advantage of a gene for cancer? And the study called removing a gene for a cancer. Mice. And most of the resulting mice died of cancer. But some of them showed remarkable longevity, which is really interesting. The tendency for something, a gene, which makes your cells grow fast, might have a value. It might be hard to control it, but it has nothing to sometimes.
Well, in fact, that's the very definition of a cancer gene, is a gene that is absolutely essential for life and for growth at some point. But when it's expressed at the wrong time, it leads to cancer, because otherwise it would be selected against. So in fact, cancer genes are some of the most important functional genes in the body.
But you need
Which needs to be controlled. It needs to be
Yeah, it
controlled.
needs to be in the right amounts, and sometimes it needs to be in the right cellular compartment.
at the right time.
Yeah, that's
the
a
right
good
time.
point.
Or expressed on the fetally, or in the first couple of years, or in life.
Yeah.
And it's just expressed
They're
in a long
getting
time.
longevity out of taking the brakes off and not quite.
Yeah.
That is interesting, but I think it is mostly that the cancer cells, like RAS, KRAS especially, it's an essential cell. It's an essential protein for your cells, and it's just you get it overexpressed.
Okay, questions? We are running out of time.
What's the most recent basis for this research on stem cells, being able to use them to create more stem cells?
Oh, wow.
They just did that research with the people who had heart attacks, where they had, most people with heart attacks have horrible scarring of the heart, and so they took stem cells from the heart of those people, they cultured them in the lab, they injected them into the scar tissue in the heart, and it healed a great portion of the scar tissue and made it into healthy tissue again. So that was really positive. Whether it's going to reduce their chance of future heart attacks, we don't yet, but it seems promising. Generally, it seems like the embryonic stem cells are less useful than the more developed stem cells because the embryonic ones easily turn to cancer. And a lot of people where that happens.
Well, the other thing, that's something else. But the other thing about stem cells, you're right that embryonic ones usually have to be a different personal genotype. Whereas if you can get stem cells from your own body that are also evolved to be similar function, then you don't need to take all those anti-suppressive drugs. But it's important to remember that the techniques we use to study adult stem cells were derived from embryonic stem cells. Okay, so the research needs to go on with that, but it is very exciting that, as you say, that if you can have stem cells from your own body, you don't realize that our body is full of embryonic stem cells in our.
And one lovely extension of that, especially when we look back at the clones that are in the description, we're probably a long way from being able to clone people. But the pathway toward that does work with the stem cells, and a really interesting side effect, especially when you consider some of the extreme political reactions to the idea of cloning and stem cells, or what can we use in embryonic stem cells, because everyone has a potential human being. We reached the point where we can reduce the stem cells, which gives us and we want to live into something capable of being please don't plug your phone, you're going to be recording. We have to quit. They're opening the door, trying to throw more people in here after us. Thank you
Thanks
very
a
much.
lot, everybody.
It's one of those stuff.
Okay. Well, I'm gonna check this guy out.